We live in a world with an aging population who want to have a fulfilled and happy lifestyle. We live in an era of chronic diseases with unmet clinical need. CSIRO seeks to provide solutions to these problems by developing materials and devices that can address some of these issues.
Traditionally, the drugs used to treat cancer patients or patients with infectious diseases are difficult to deliver to specific targeted cells. So we're working on developing new polymers to help carry these molecules into target cells to treat these patients. We have made very good progress so far. We have tested these materials in the lab and also now moving in to test them in animals. And we believe that these new materials will help the cancer patients and patients with infectious diseases for them to actually have a better treatment option than the currently available treatments.
Scientifically, one of the really exciting things about this area is that it brings together multidisciplinary teams in material science and cell biology to tackle some of these problems.
The multidisciplinary approach has been very useful in the development of a fibre optic catheter for measuring colonic motility. The basic technology started off in the telecommunications industry, which is my background, and then coupled with the material science expertise at CSIRO, and very importantly, the clinical expertise of my colleagues at St George Hospital and Flinders Hospital in Adelaide, has allowed us to develop a tool, a diagnostic tool, that first of all does what it's supposed to do, but also very importantly does it in a way that is acceptable to the clinical community, to such an extent that recently it's been referred to as the Hubble telescope of gastroenterology.
We're in a new era where materials are now helping the body to heal itself.
In Australia at present, there are 310,000 people who inject insulin on a daily basis to control their blood sugar levels. The Diabetes Stem Cell Project aims to replace the need for such injections by transplanting stem cells that have been induced to make insulin. And in order to protect the cells from destruction by the recipient's immune system, we place them in these microcapsules which are made out of a product from seaweed called alginate. Little pores on the surface that allow insulin to go out, allow nutrients to come in, but are too small to allow the recipient's immune cells to attack. In time, we expect that the need for insulin injections will be replaced completely by this type of therapy.
Right now we have a major problem in providing enough blood cells and platelets for transfusion purposes. This project is all about making those cells from stem cells. The main reason for doing this is there are patients that need platelets. For example, people that are being treated for cancer and or receiving bone marrow transplants. There simply isn't enough platelets from normal donors to provide what's required for these patients. So we're about making these platelets from stem cells in the laboratory. Right now we can do this at laboratory, small scale, and generate functional platelets. The real challenge is to increase the scale and produce enough platelets so we can treat people that need them.
So with the idea of large-scale manufacture of stem cells in mind, the way we're approaching it is by using synthetic materials, embedding biological information in them and using high surface area so that we can generate large quantities of cells for therapeutic use. One of the reasons for using synthetic materials is so we can avoid the use of animal-derived products, and using this approach we can hopefully revolutionise the manufacture of stem cells for therapy.
Ultimately for me, the thing that really excites me and motivates me in this area is bringing together multidisciplinary teams that tackle unmet clinical needs and to provide solutions through provision of new products, new devices and new materials that are going to make a difference to everyday life.